Good morning, everyone. I'm just watching the participant numbers climb as people are being led into the webinar, and we'll get started in just a moment. I guess while that's happening, I'll just introduce myself. My name is Dr. Jeffrey Miller, and along with Dr. Kate Elkington and Dr. Christine Denny, I'm one of the new co-directors of Grand Rounds in the Department of Psychiatry at Columbia and NISB, and I want to welcome you all to Grand Rounds this morning. We have a wonderful Grand Rounds today, a topical Grand Rounds, which is the Developmental Psychobiology Grand Rounds. I just want to make, we don't have significant announcements, so we'll be able to have plenty of time for our talk today. I just want to make one comment about questions. So we welcome everyone's questions, and you can pose questions at any point during the speaker's talk today. I'd ask if you hover toward the bottom of your screen, you should see a Q&A button, and I would ask that you use that Q&A button and not the chat button. That way, all the questions will come into the same place, and it will be easier for the moderators to see them and to see your question and to transmit it to the speaker. And another point about questions is that we have a new policy that we are initiating of trying to prioritize questions from trainees, including students. And so if you are a trainee and have a question, we would welcome your asking it. And please put the word trainee at the beginning of your question in the Q&A feature. So with that, we'll get started, and I'm going to hand the virtual microphone over to my colleague, Dr. William Pfeiffer, Professor of Medical Psychology in Psychiatry and Pediatrics here at Columbia, to introduce today's speaker.
Thank you very much, both I want to thank the organizers for allowing me to first learn how to retie my tie. Haven't done that in a long time, so I like that opportunity, but also to be able to introduce our distinguished speaker, of course. Moriah Thomason is the Barakett Associate Professor of Clinical and Adolescent Psychiatry and Population Health in the NYU School of Medicine. She formerly served as the Director of Perinatal Neuroconnectivity Unit within the Intramural Perinatology Research Branch of NICHD and NIH. She received her undergraduate training at UC Berkeley and her graduate and postdoctoral training at Stanford and MIT in neuroscience. Her work has been featured all over the place, including NPR, All Things Considered, BBC World Service, Huffington Post, MIT Technology Review, New Scientist, and most recently in Science, Nature Medicine, and National Geographic. In 2019, she received the honor of the Presidential Early Career Award for Scientists and Engineers from the Office of the President, not the President, from the Office of the President of the United States. And the impressive breadth and depth of her research addressing principles of neurodevelopment beginning in utero, along with very recent funding news, is just in the last few weeks with 2 more national cohort studies. She'll expand even more her team's ability to uniquely investigate environmental factors which can influence the functional neurocircuitry of the developing brain. So it is my distinct pleasure to introduce this year's Developmental Psychobiology Grand Rounds speaker, Dr. Moriah Thomason. Moriah.
Thank you, Jeffrey, and thank you so much, Bill. I'll go ahead and share my screen. It is a pleasure to be here. Just highlighting the fact that I'm a neighbor to you all. So if there's anything interesting, anything you want to touch on more, any questions that are outside of the scope of what I'm discussing, please, I hope that you'll reach out in the future because we can eventually maybe even meet in person. Really the focus of my talk today has to do with signatures of maternal stress in the fetal connectome. And just to get a little bit of background on me, I'm trained as a neuroscientist, and the primary objective of my work generally is to understand fundamentals of brain development and how the growing brain is impacted by the environment over time, even before birth. And to achieve this, we collect multimodal data, biological data, imaging data, electrophysiology, and in our cohorts, we prioritize economic and racial ethnic diversity and take a longitudinal approach. Altogether, what we hope to begin to do is to understand the relevance of very early brain development to the emergence of common neurological and psychiatric diseases, And today I'm going to give a little bit of background on discoveries we've made over the past 7 years using MRI in the fetal brain. And then I'm going to turn to focus on this particular topic that's important to us, which is the role of stress and inflammation to very, very early human brain development. So the primary technique that my lab has used to study the brain before birth is resting state MRI. This is something that's probably familiar to a lot of you, but quickly it's reliant upon the principle that spontaneous activity in the brain has an organized structure, and that if we extract time course from a specific region and simply correlate that spontaneous time course, so the participant is not engaging in a specific activity, which is why we use the word rest, even if the person is awake, they're not doing a specific task. We take the spontaneous time course here, the left motor cortex as an example, correlate it with the spontaneous activity across the entire brain system. The answer that we get is a perfect representation of that entire sensory motor system. So we've recapitulated a structural and functional system while somebody's resting quietly in the scanner. And so this is maybe surprising, maybe not surprising, because these are areas that are highly anatomically connected and also are very regularly co-engaged for the performance of task. And in this way, we're able to pull out not just, let's say, the motor system, but also higher order cognitive systems, other primary sensory motor systems. And we can look at interactions between them, across them, entire brain connectional organization. And this is actually a really great thing when you're trying to understand the human fetal brain development, because we can't easily ask the non-compliant fetus to complete a task, but we do want to understand these systems. So it probably doesn't come as a surprise that this is a challenging enterprise. You know, a lot of the common things that we think about are that motion is a big confounder, so we have to concern ourselves with that. But along with that, we have no single orientation for image analysis. We lack processes and methods for naming areas, for analyzing these data. Further, the fetal brain is small, so the effective resolution of the images that I'm able to obtain are lower than you would in a fully adult-sized brain. And if that wasn't enough, the fetus is encased within an independently moving maternal compartment that also is contributing noise. And then finally, we possess very limited knowledge of the physiological basis. And in this case, we're talking about functional MRI studies. So those studies are dependent upon understanding neurovascular coupling. We have an immature vascular system. There are things that we still need to learn about those associations. So if you're wondering, we have published a number of studies that talk a little bit more about the approach. I think the main thing that we need to focus on to have a conversation about this as a basis is that what we do with the fetal data is that the fetus alternates between periods of motion and periods of quiescence. And so what we do is we go through and we find those periods where the fetus remains still, and those are what we analyze. So just to give you that idea, we collect more data than is required, and then we analyze the best of those data. So that's an important and costly feature, but it does make this really new area possible. And who knows, in the future, we may be able to utilize more of the data. So what I'm going to start with is a little bit about what we've learned about normative brain development. We now have evidence that as a fetus matures, the efficiency of the overall network structure increases. So again, these are based on fetal resting state data, looking at functional signals and the time it takes a message to get from one part of the network to another. We use graph theoretical analysis in order to come up with these metrics. And what this looks like, and these are spring plots that a colleague of ours, Martin van den Heuvel, made, looking at actual fetal data, what you see is that you have these like clusters of local neighborhoods that over development begin to give rise to these more connected, interconnected neighborhoods. And that's what I'm talking about when I say greater efficiency, because it's less time to get from one system in the network to another. And this makes sense because we know from other studies, and this is just a schematic of physical development, that physical outgrowth moves from being local connections to more increasingly distant brain regions across fetal development. We also find that modularity decreases with advancing age, and this also fits with that efficiency story. Again, those subnetworks are given different colors here to represent what regions they span. And what you see across fetal development, just to describe it for a moment and not focus so much on the image, is that these networks become more interconnected with one another, which means modularity or independence or separation of these networks becomes less so. So you're seeing more crosstalk between networks. And this is in contrast to what we see across childhood and adolescence, where what you see is a shift to more independence of the subnetworks of the brain. So we see less crosstalk, we see increased modularity in that adolescent transition, which is interpreted as the increasing specialization of brain systems. So you have, say, a brain region that's really important for reasoning, that is going to become potentially less interactive with regions that don't support that process. So that is sort of what I'm describing here is somewhat like an inverted U, where we see increasing connectivity very early in development, crosstalk of networks, pruning, refinement, and specialization, which makes sense. Now we're just able to measure it, and I think that's what's exciting about this work. We've also learned that the fetal brain has hubs in terms of the total network architecture. So like central train station, Grand Central Station for the brain network. And that overall the fetal brain has a rich club organization, which means that highly connected regions of the brain or hubs are preferentially more connected to other highly connected regions. And this rich club phenomenon increases across fetal development, which is shown here. So overall, what I've described so far are topographic properties that suggest the existence of a dense and sophisticated interconnected network structure. The studies I'm talking about today are studies that we did predominantly in mid-second trimester to third trimester. So we're talking about weeks 26 to term. And what we're seeing are principles that really do approximate what we see in a mature brain, which, to be fair, could have been a surprise to us. We didn't prior to this technology, didn't really have a basis for coming to conclusions about how regions are communicating with each other or how much those look like what we see postnatally. So having observed these features led us to want to ask, how much overlap exists in the entire organization of fetal brain to, say, an adult structure. So what I'm showing you here is a group of adults where we took the top 10% strongest connections across the entire brain, shown here by blue dots. We sorted them by neighborhoods they reside in, sort of a kind of fingerprint of who's talking to who. And what I want to ask all of you, although I know we're in a webinar and you can't respond, is if we were to perform the same analysis in a group of fetuses, how similar would that total connectome architecture look, where 100% is complete overlap and 0 is none? And when you think about this, you probably ought to cut that number down quite a bit, because even though I've shown you some similarity in organizational principles, say compared to a random network, we still have a lot of noise represented in these data. We can't take the noise out of MRI data. We can't even take scanner variability or individual variability out. So how much do we see overlap in a fetal and an adult brain connectome? And the answer is a striking 62%. So now I'm going to turn, I've sort of given you this primer on the fact that the fetus possesses an elaborate network architecture, which naturally leads us to examination of programming by prenatal stress. So in a recent study, we sought to determine whether the stress of a mother was related to fetal neural development. Now, prior studies have confirmed a programming role of prenatal stress. Much of that work has happened here at Columbia with the beautiful work of Catherine Monk and many of you who might be here. And what we do know is that beginning in infancy, differences in behavior and brain development have been characterized and related to that prenatal programming. And we know that these effects seem to be long lasting. So we see them across the life course. We've also seen an association between positive affect and longer gestational course and negative affect and shorter gestational course, and also more birth complications. A couple of notes about this is that the association between psychological health persists when controlling for the usual suspects, and also this association seems to be stronger later in pregnancy. And also for those of you that are beginning to ask in your mind about mechanistic pathways, We know that inflammation is one of those proposed intermediates between maternal stress and offspring neurodevelopmental risk. And we also, more recently, there's been a lot of consideration about the role of mitochondria function and potentially bridging between that inflammation and outcome association. But much of our understanding does arise from preclinical and also postnatal studies. So our primary goal in this study was to begin to understand these complex associations in the human brain and to do so before birth. So what we did is we evaluated functional connectivity in 118 fetuses. Inclusion criteria for this analysis were that data were already processed and analyzed, because in our lab, there's a lot more collection. Processing is so manual and takes so much time and quality checking that we don't have all of the data available. So that was a parameter. We also did exclude on the basis of being born very preterm or having birth weight lower than 2,500 grams. The approach that we used was a very data-driven approach that allowed the functional signals of the brain to divide the brain into small parcels or regions. So we used a data-driven approach for dividing the brain up into regions of interest. And then we applied a community detection algorithm to further divide these individual parcels into neighborhoods, or more precisely, networks. And what we found was that the stress of the mother was associated with differences in frontal parietal and striatal connectivity. And I want to spend a little bit of time thinking about these particular observations in these particular areas. First of all, we know that these are connections that are forming prenatally because we have normative studies that have shown that to be the case. Also, we know that these are regions, these are connections that are modified in response to trauma and even in response to acute stress. The frontal parietal network in particular is implicated in a number of diseases, so much so that individuals, especially in the adult literature, do refer to it as possibly being a transdiagnostic observation. And then finally, we did have a hint that these were right lateralized effects. And again, this is us using a data-driven approach, so we're not going to overinterpret these findings. But suffice to say that having observed a more right lateralized effect does seem to match with some of what we see in the animal literature. So that's something that we might want to come back to. There was another observation that we made, which was differences in frontal, motor, and temporal insular systems. Again, these are regions that we know are present and are developing across fetal life. So they are candidates for seeing differences that are associated with maternal stress. I find differences in the insula particularly exciting, and also temporal regions, because these are areas that have been identified as hubs of connectivity. Furthermore, studies that combine EEG with MRI and also animal studies have shown that these regions, the insula and these inferior temporal regions, are important for generating spontaneous signals or firing during prenatal life and in early development, which could suggest that they're instrumental in neurogenesis and also important for circuit building. So it's possible that prenatal variation in subnetwork functional connectivity that we see here predisposes these children to long-term processing differences across these and other related behavioral domains. So this sort of raises important questions about future development, but a challenge to findings such as these are that, and this is, sort of the double-edged sword of research in this area, is that there are a lot of different factors that may have potential to influence prenatal brain development. And it's hard to control for all of these factors. So we're confounded by these factors, but why I think of it as a double-edged sword is because if we can identify and isolate what these factors are or how these factors may interact with that stress to outcome association, we may be able to identify what is predictive or begin to understand what are the processes that are important that underlie this. And if we can identify what's predictive, we could potentially push the needle and help those that are impacted, which is a significant swath of the human population. So to begin to get at this in the present study, which was reasonably sized at an N of 118 to look at interactions, is we looked at health, lifestyle, and emotional support as possible factors that may interact with this stress to fetal brain relationship that I just described. We use factor analysis as a means of reducing the number of variables. We put these derived factors into our models and found that a modest significant influence of this particular factor, which I will come into in just a moment. And the nature of this association was that it almost seemed that the women that you might characterize as having less good health seem to have less impact of the stress to brain association. So less strong association in those women shown in the dark dots that had less good health behaviors overall, more impact of stress in our participants that seemed healthier overall. So it was almost as if the systems that impacted the fetal brain might have been a little bit more flooded, and that there was less movement in those for the women that had greater stress. And these were the aspects of behavior that informed this particular factor. And we did find that there was also variation in how much each of these factors contributed to that brain stress association, sort of amplifying opportunity for intervention. In particular, we really see that sleep is an important driver of this association. So we did take this work one step further and ask whether the degree to which the stress of the mother was reflected in these changes observed across the group was related to the child's age when delivered. So again, we talked about this a little bit in the introduction of this talk. We wanted to know if the stress brain effect predicted how early a child was born. And we found that it did. So in other words, the stress on the parent raises the likelihood of premature birth in addition to potentially modifying the baby's brain. And since we already know that being born early sets the child at a disadvantage, this baby now has two challenges. Now, I do want to take a step back and say that it's not, I don't think that it's that the degree to which the brain was impacted by stress caused and was causally related to preterm delivery. I think instead what we have here is a common genesis of two different types of outcomes. But being born early does have its disadvantages. So this is something that we need to think about more closely. So in closing, I do want to place a few conditions on some of what I've shared today. First of all, the effects we see of psychological stress during pregnancy are modest. So I did give a little bit of background when I set this up and talked a little bit about stress explaining differences in gestational outcomes. But Pessinen, for example, one of the studies I cited did show that this effect, the size of the amount of variation that stress explained, it was anxiety in that particular case, was 1.1 to 0.2%. So we have an overall small effect size potentially in the kinds of studies that you'll see around this. However, having said that, a large population of women experience mood disorders and stress during pregnancy. Depression in pregnancy is estimated at almost 1 in 5 women. So I think what I want to communicate to the group is that even if we are talking about rather modest effects, even if there are environmental considerations that we need to take into account, such a huge amount of the population are impacted by this condition. So we really do need to think about this as a significant cumulative societal effect and one that requires a lot more work. And in terms of next steps, our group is working to understand the relevance of these prenatal developmental differences to developmental variation in childhood. And the way that we do this is that, as I mentioned early in the talk, is we follow children over time. In a recent study that I'm not going to share with you today, just to not overwhelm you with too much data, we looked at the effect of cannabis use during pregnancy on fetal brain network development. And in that study, we did begin to have outcomes at age 5. We didn't have enough age 5 outcomes to do the perfect model that we really are working toward, which is prenatal predictor, intrauterine brain predicting age 5. But what we did is we took all of the cases that we had age 5 outcomes, all of the cases that we had the THC exposure for. We created maps about hippocampal connectivity at age 5 as it was related to more advantageous outcomes, better behavioral outcomes at age 5. And then we use those age 5 maps, brain maps, to filter the effects of THC. That way we could say, okay, if the THC group has more connectivity, is that in region A, is that connectivity related to less good outcomes or better outcomes? And we were able to filter that data in that way. And what we found was that in the THC groups, connectivity was associated with less good outcomes. So we could be a little bit more concrete about the interpretation of our findings. And that's an important feature because in field work, we don't have a lot of priors in which to contextualize a particular connectivity difference that we might see. And maybe that's something that we could talk about a little bit in the discussion. The fetal brain is really open territory. And so we do have to be careful when we think about networks as serving the same functions that they do later in life, because there's not a great amount of evidence to suggest we should. So in terms of what we are doing now in the lab is we're studying markers of inflammation in order to better understand some of the mechanistic linkages that I discussed with you before. We're doing this in placental specimens and also with markers in the teeth that children lose later in development. So we know now that teeth are important biological hard drives of the exposures that children have had in utero. And in fact, there are studies looking at teeth in twins showing that even within a twin pregnancy, the amount of, say, toxicants, let's say lead, that you can detect in the tooth material can differ from fetus to fetus, suggesting that this is a very nice representation of what is reaching that fetal system, as opposed to taking something from the maternal periphery and having some idea. You really have a direct measure in the fetus. So we've been collecting children's teeth that were scanned in utero for more than 4 years. And we now have some funding that will allow us, with collaborators at Mount Sinai, to begin to take apart things like micronutrients, like inflammation, and begin to think about what are some of the things that may underlie these differences that we see in the brain. I also have the pleasure of showing this new baby scientist. This is Rowan, born 5 days ago, happy and healthy. And many of you might know Uju Berry. So this is Uju's new baby. And I just wanted to demonstrate that she's forming her teeth. The teeth, just for those of you that aren't familiar with this, begin to form at week 15 of gestation and continue to form through the first year of life. So I think this is a really promising inroad to begin to try to understand in a human model, what are some of the mechanistic pathways that underlie these important associations. And I hope that you're convinced that this is a significant area for research. So there are a lot of activities happening in the lab right now. I've sort of focused on developmental disease origins and some of our ongoing work. Much of my work is actually based in a cohort that I established in Detroit beginning 10 years ago. The oldest children are reaching age 9. I also, for any of you that are really interested in technology, the technology itself, the fetal imaging approaches, I want to just highlight this workshop that's happening this week. For any of you that are interested, it's a satellite event to the Mackay workshop. This is a really important place for sharing the processes for analyzing these very challenging data. I hope that were I to come back to talk to you in another 5 years. And I know beautiful work by Dustin Scheinhaus and Marissa Spann, who many of you know, are really kind of, as a field, I think we're all kind of working together to try to figure out how to do this research better. So data sharing methods is extremely important. On the bottom left, I'm highlighting the COPE study. The COPE study is an interesting project that arose out of the peak of the beginning of the New York first wave of the pandemic in March of 2020. Researchers from around the world came together and they shared common data elements and started cohorts all around the world comprised of pregnant women, new parents, and even older children. And that group has continued to communicate and has given rise to the CoVGen Research Alliance, which, if you're not familiar with it, if you're interested in these topics, if you're thinking at all about how the COVID pandemic psychosocially or physically is impacting development of the next generation of children, I really encourage you to go to this website, potentially reach out, potentially become involved. It's a completely open network of researchers around the world that are going to sort of, it's a bridge basically to build connections. And we are hosting our first international summit. It's going to be a satellite event to the ISDP conference that happens in November. So again, please look it up. I'm tremendously excited that this is happening. Bill sort of mentioned for just a moment that that an upcoming major initiative of the U.S. is to do is focused on the healthy brain and child development study. We are going to be one of the participating sites in that initiative. There will be 25 across the United States. This is, for those that don't know, this is like a baby ABCD study. So if you're familiar with the child, adolescent, and brain development study, this is an initiative that follows on the heels of that to begin looking even earlier in development, beginning prenatally, following children for at least 10 years. And then finally, just last week, it was announced that there will also be a national effort to study more than 20,000 individuals across the United States that are recovering from having been infected with COVID, looking across multiple domains, including neurological, but also different types of clinical development to understand potential long-term impacts. So I'm going to thank you for your attention. Also thank a wonderful lab that works on this. My closest collaborators on the studies I've talked about today are Natalie Brito, who is my co-PI on the New York cohorts, and then Chris Trentacostas, who's in Detroit. And I do realize that I've ended quite early. So what I need to do is, at least before we open things up, seed the fact that we do have a lot going on in terms of recruiting women who have had COVID during pregnancy in New York, also children that have been infected with COVID, and even looking at COVID in adults. And so I just want to sort of seed that in case, because that isn't something that I've discussed with the group, but if it's something that is of interest, it's something I can easily talk to you more about. So with that, I will stop. And I'm sorry my talk was a little bit shorter than planned. I had thought that there was going to be a ramp up. I thought that I had a shorter duration to talk, Bill, so I'm sorry I ended early.
No, we lied to you, Moriah. This is great. And yes, it makes me think I wish we could spend another half hour or more on this. It also made me feel like what we're also really missing is, besides the in-person aspect, one of the nicest parts of our grand rounds is to be able to sit afterwards and have the trainees come and be able to ask you very specific questions and really kind of get into the weeds. And it's so hard to do, you know, with this large group, amorphous group out there somewhere that we can't even see. So first, I want to thank you for that. And two, I would like to give you time maybe to go into what you were just alluding to, but there were a couple of questions that made it through the chat and then onto the Q&A, if I could give them to you. One was how, it's relevant to what you were just saying, how are you able to continue your assessments during this pandemic, and how are you able to feel, you know, that you're, in fact, has it helped in being able to get more information that you might have not have been able to get before remotely?
Yeah, I just wish I could just open up my computer and show you guys, because I feel like there's so much more I could have put into the talk. But yeah, I think there are two things there. One is, how have we innovated? And one is, how has science had new opportunities to address questions in different ways? That's what I'm hearing. So to the first, there's been just a ton of innovation in remote assessment of infants. And in fact, we do have a paper that's just coming out in Developmental Cognitive Neuroscience that kind of walks through some of the methodologies that we developed. I think amongst the most interesting are, how do you collect biological materials remotely from families in their homes? And also, how do you assess infant development over a Zoom? And so just to speak to those quickly, and I'm happy to share materials and processes for anyone that's thinking about doing this. I think it's important. I really do. It doesn't have to be that you're studying children. It's really also for any kind of biological specimen collection that you want to do, minimizing burden to participants and coming in to see you in person. We're able to collect blood, breast milk, hair, nails, saliva, and stool all remotely. And so if you want to go on our website, Baby Bees, like a bee that flies, BEES, we even have like explainer videos for participants about collection at home. We also are using a device called Tasso that many of you have heard of. Seventh Sense, Tasso, Mitra. We explored all of these different devices for collecting blood remotely. So even better than doing a finger prick, you can use this device. I actually have one here, so maybe I'll show you. But you can use this device. It simply sticks on the skin. You depress a button. It's a quick lancet in and out. Because it's a sticker, it creates a vacuum and the blood can passively drip into a collection tube. The participant snaps it off, screws the top on, puts it in a pre-labeled, pre-everything box, insulated, cold packs, and sends it to the lab where we extract serum when it gets to us. It's complicated is the answer, but it is working really well, and some things are very easy to collect. Other innovations, I have a postdoc in the lab, Denise Warchan, who has quite remarkably developed an algorithm for recording or for analyzing infant eye movements during an attention task over Zoom. So I'm not getting into all of the various pieces. Again, I would point you to that paper that we've just published, but we're able to measure infant attention and do eye tracking over Zoom. So lots of innovation, I think, in the types of things we can do, which is very important for a lot of the things that a lot of us think about in terms of reaching hard to reach populations, because it's not just a pandemic thing about families not being able to come into the lab. It's a urban communities things. And Bill, you demonstrate this very well in your work. We need to be able to get to large parts of the population that aren't able to come into research institutes for studies. So the more we can do to continue to innovate in remote data collection is critical. Can't quite take my MRI machine to houses, but we do have some funding right now to put a hyperfine microtesla MRI machine into a van and drive it to people's houses. So we actually are also beginning to innovate and take neuroimaging labs to people's homes. So that's an exciting thing that's also happening. In terms of innovating in science, a lot of folks think about this as a quasi-experimental design. The pandemic as a force that cuts across all social economic strata is something that creates a different experimental design than recruiting women and looking at stress during non-pandemic times. So that is something that we've seen illustrated well with historical examples. Even the beautiful work that's come out of Colombia on her Superstorm Sandy, and the work that that group has done has been really important for understanding prenatal effects. So long answer is tons of innovation happening as a result of this pandemic in early life assessments. Conclusion number two, I think some of what is happening is going to help us get better representation in the research that we do. And number three, it does provide a different type of experimental design that can address some of the causal factors that influence these studies.
So not surprisingly, there's a number of other questions here because people are so interested in this wonderful work you're doing. So I was asked to read a couple of the trainees' questions first. One, I can't see her last name, but Hannah S. says that she's curious in the fetal exposure study how you were able to control for environmental stressors that may be correlated with cannabis use, and also were there dose-dependent effects observed in the in brain development and connectivity.
Anna, I'm really happy to share that paper with you because it's going to do a better job than I can do, but I can address this. It's a huge, huge topic. And I looked at the literature when I performed this analysis, and I want to tell you, I work with women. I didn't get into the sample very much, but a lot of the women in that study are socially and economically disadvantaged. It's a predominantly Black sample in Detroit. They are highly stressed. They have a history of trauma. They have intergenerational risk transmission. This is a community of people that are stressed. And I'm going to be completely honest and say, I wanted to be very agnostic to the idea that cannabis was bad, because these are women, some of whom may be experiencing some therapeutic benefit from having cannabis be part of their pregnancy. So it was important to me to come in with a neutral lens, because a lot of the studies that I saw out there seem to just be coming from the standpoint of, if you're using drugs, that's going to harm your baby. We don't know if it's drugs that are really going to have a big effect size on brain development, or if it's something else. So Anna, your question is really important. To make those kind of attributions it's pretty heavy when you actually meet these ladies, talk to them, and realize that you don't want to scare them unnecessarily. So I did look at that. I looked at the literature, and I don't think that we've done a great job as a field. I did perform these analyses. I'm trying to remember what exactly the statistics were. Stepwise analysis, where in our regressions, what we did is we took out these significant areas that survive very stringent threshold correction, multiple comparisons correction. We had 6 models where we had these significant effects. We added to those anything that differed between our THC group and our not THC group statistically. The things that differed were anxiety, stress, smoking, and alcohol. We put those things into these models in these stepwise fashion and looked for every single region. Was the brain effect related also? Did those factors additionally explain variance in those? Did they improve the model essentially? And for many of the regions, they did. And the 2 things that had the biggest effect were anxiety and smoking cigarettes. So again, the major takeaway from these studies, and all of the studies you'll read that won't do those analyses, is that it isn't just a cannabis effect. It isn't just about the cannabis. It's also about these other things. And I think that that's important for not, I don't want to say shaming these women. A lot of women are just dealing with a lot of really hard issues, and causing them additional stress, if we can't really make these causal attributions, is something I'm very cautious about. So we have to consider these in the model. So thanks for asking.
And there's a related question, Moriah, from Meg H, who says that she wondered, in your measurements of maternal stress, how confident and how do you deal with mothers that information they might not want to reveal? Specifically, she asks about drug use, etc.
Yeah, no, that's a really good question. There are a couple of approaches that we've used in our work. One is we do also have biological measures of stress, so we can look at whether or not there's correspondence between cortisol measured during pregnancy. That's one approach, right, is to take biological measures. We also have access to medical records, so we can do sensitivity analyses within those women that we know have positive drug. That's actually how the THC study was done. It was not based on self-report. It was based on urinalysis. So I guess the short answer is that the biological materials, even if they're not available in all of your cases, can be used to perform sensitivity analyses or to look at whether there's some reliability in the self-report data that you do take. The study that I talked about today that assessed stress, we did find a significant association with cortisol in the subset that had both available. Also, we used a factor score. I didn't get into it in this analysis, but I really struggle with which predictor, which self-report variable do you put into a model of stress? I mean, stress is this incredibly giant construct. So what we did is we took perceived stress, we took anxiety, we took depression, we took worry, and we took, there was a 5th variable. Might have been satisfaction with life. If we took these variables in a factor analysis and used that factor as a predictor, and then we also published the loadings of each into that factor, so that as a reader, you can sort of take away, what was this predictor? What does it mean? But very good question about, how do you define these things? What reliability do you have for these things? What are the other things? We spend a lot of time with women. These are really complicated things to measure.
Another trainee question from Aisha Sanya asked, you had mentioned before you also study the other suspects that were related to whatever your primary interest is. So for example, is there something about how strongly smoking might be related to cannabis use? And I think probably part of that is, other than their co-occurrence, the fact that people are taking in their nicotine in many different ways. And is that something that you are considering in your studies?
Yeah, it's not as something that I'm particularly focused on, but there are others that have really probably much more. A study that was designed to look at smoking would be designed differently than my studies, because it's a kind of a convenient sample of smoking. In my case, I'm not recruiting on the basis of smoke use. I'm not documenting very rigorously the patterns of use or the kinds of use or the kinds of tobacco or the combination of different types of things you can smoke. So I'm not the best to say that. Yeah, you really would have to design a study. Fortunately, the literature on smoking is quite large, and even, meaning we know more in that area than we know about a lot of newer things, even more than THC. We know smoking has a huge effect size. So I think one of the things that I'm thinking about with the coming HBCD study, which is largely focused on opioid exposure during pregnancy, is that at the end of the day, after this huge, I hope no one from NIH is here. Okay, maybe they are. At the end of the day, I think when we look, and I'm interested, Bill, how your thoughts are on this, if we look at effect size of opioid exposure compared to cigarette exposure, which are going to be highly correlated, at the end of the day, cigarette exposure might have a much bigger impact on brain development than opioids will. There are some people that believe that that will be the case.
Yeah, that'll be. I agree with you. If we're not looking at least at smoking and the related suspects, and we might miss the boat on the answers to these questions.
Yeah, we found that with HIV.
Yeah,
all those years ago, that HIV itself wasn't harmful to these babies, even crack.
Yeah.
It was the social environment that was having such a tremendous influence.
Let's see, Ezra Susser has, I think, two questions I'm going to try to combine into one. One had to do with COVID in adults, and I think he specifically was thinking about the pregnant women. So the long hauler study that you're about to engage in, is it going to be divided between moms, pregnant women, and the moms, and the children? Yeah,
there's a planning meeting happening right now. So we're still working out some of the details. But yes, there will be separate pregnancy cohorts, and in those cohorts, there will be additional biospecimens, including staining the placental material for COVID virus or SARS-CoV-2. And in fact, there's beautiful work that comes out of NYU Langone already showing that the virus is present in the placenta. So there's a huge amount of research to be done just in pregnancy. And I believe at Columbia and Mount Sinai, groups have been pursuing some of those questions as well. So I'm certainly not alone. I'm in good company, that we do want to try to understand that better. We have a lot of neuroimaging data already, and other groups I think are working on trying to understand infection during pregnancy and the impacts on the fetus. I have to say I'm really optimistic that the effects are incredibly small and that these babies are going to be just fine. And I really hope that as a field, we are just as brave in terms of publishing null findings and non-significant results as we are quick to run to publish negative findings.
No, that's important.
It's a really important point. And I've written a commentary and submitted it to a journal because I'm fearful. I have families that are in our COPE study now who come to their appointments and ask the RAs if their babies are okay. Then these are not just the women that had infection during pregnancy. These are just moms that are hearing through the news that their children might develop differently because they were born during the pandemic and maybe didn't have as much social interaction, and parents are afraid. And so, as a field, I think we have to do a little bit of self-policing. We have to do pre-registered reports. We have to think about effect sizes. We have to compare the magnitude of these effects, and we have to be really cautious about our messages. I got a little bit on my soapbox just then.
I'll join you on that soapbox, because I do think that another way to frame that is to tell our parents we aren't just looking for these negative adverse effects. We're looking for dealing with questions around resilience and positive health, which are also part of this. So it isn't just how bad it's going to be, but let's think about these other questions. So I think it's tremendously important. I forgot to mention part B of Ezra's questions was, could epidemiological studies of populations who experienced acute disasters historically help estimate effect sizes, especially with the severity of exposures that are for
I
individuals.
absolutely hope so. That would be one of the most powerful ways to use this situation. I really think so, because then you can compare. There was this great study published by, I think it's Orion. If you Google potato, I'm not kidding, and screen time, you'll find this paper that shows that there's an effect size of 0.4%, or explaining the variation of screen use predicting adolescent outcomes. So more screen use, less good outcomes. But then the authors of, I'll call it the potato study, just so you all remember, go look it up. They also looked at other negative potential impacts, neutral and positive, and they found that the magnitude of the screen effect was on par with how much you listen to music, how tall you are, and how many potatoes you eat. And so this is really important, because we are publishing papers and we are not enough publishing effect sizes. And there could be things like eating potatoes that are similarly influencing these. So the epidemiological studies really give us a shot at that, which I really appreciate.
I thought for sure you're going to go with couch instead of potato on that, but wow, that's really interesting. Cristiani Duarte asks, first she says, great talk, thank you so much. Can you expand on why the relationship between maternal stress and offspring connectivity is weaker among women with poor health? Do you expect the same among your COVID plus moms?
Yeah, I think it was Christine. I appreciate that question greatly. I've thought about it a lot. I thought about it in terms of animal systems and also human behavior, because it wasn't exactly what I predicted, to be honest. And the best conclusion that I came to in terms of what might be happening here is that it's almost a ceiling effect, like almost like those same circuits that are being hit by sleep or lack of sleep are the circuits that are first to be impacted, which kind of makes sense if you think, if we were able to go down into the animal and be more mechanistic about what are the chemical differences of lack of sleep, what are the chemical differences that are a lack of or increased stress. Again, thinking about inflammation, thinking about mitochondria, we might naturally come to the conclusion that the areas of the brain or the circuits or the processes that are susceptible may have overlap, and maybe in a way, I mean, this is a really simple and reductionist idea, is that those systems are already somewhat saturated, so there's less movement. So I don't know if that's the best explanation. It's easy enough to try to think about it that way, but there might be more.
By the way, you remind me when you mentioned sleep, as people in our division, in particular Maristella Lucchini, is very much interested in finding better ways to assess parameters of sleep that are more than what we get from questionnaires, and to be able to actually send information out with wearable devices that give us more than just even sleep and activity, but a little bit more about what's going on with the underlying physiological rhythms that are determining how well you sleep and for how long, and parameters of sleep health. So yeah, I think that's going to be it.
Yeah, we've actually done a study where we looked at maternal childhood trauma as a predictor of sleep behavior, and we had the kiddos in that same Detroit cohort wear actigraphy, and we performed a wavelet analysis that allowed us to look at sort of the rhythms of sleep, and saw significant associations that were also related to EEG alpha asymmetry. So I think what you bring up is, you know, we used to do sleep in sleep labs, and that's where we got really sophisticated data. But now with advances in engineering and informatics, frankly, because how do you process these data? We have an opportunity to really take apart sort of what it is about sleep in the brain, which is really a frontier. Yes. So yeah, wearables they're kind of doable, so people can do them.
Wearables, yes, no, yeah. That's right. Let's see, William Tucker asks, would it be fair to say that pruning, both pre and postnatally, is a major route by which brains individualize?
I think so. I really do. I know that's a short answer, but I do. And I think, you know, it's a use it or lose it kind of a system, kind of like a muscle that would atrophy if circuits aren't being used, if they aren't being deployed meaningfully. There's this there's a system by which they know that they're not needed. And we think of a lot of disorders of development as being disorders of pruning, right? That pruning can go awry. It's not just I used it correctly kind of a situation. There may be some underlying genetic disposition that changes how efficient or well or how that pruning process takes place.
Let's see, Catherine Monk asked the question. Actually, we must be related because it's similar to the one I wrote down in case we didn't get enough questions. And she echoed a similar theme, which is wonderful talk and work. Thank you. Could you speak to sex differences in your work, which show up so much in the prenatal programming work? Of course, particularly in their effects on different timing and manifestations of psychopathology.
Yeah, we have not, we've published one study looking specifically at normative development, male, female. So direct, more of a neuroscience, basic neuroscience paper. We haven't specifically taken apart differences in males and females in one of these more, these bigger models of risk. So whether it's stress to brain, whether it's child outcomes, we will have the opportunity to look at those factors, but we haven't in these particular studies.
All right. Ralph Wharton asks any conclusion or conclusions, let's see, on the meaningfulness of eye tracking data.
Oh gosh, I am not the best person to answer this question, but yes, and in fact, well, I shouldn't say yes. Infants are not easily assessed. I'll just say they're able to do less for you, right? But the eyes as an observer, and we get a lot from parent reports. So there's some circularity that all of us appreciate. And maybe because many of you work in the adult sphere, it probably comes up for you in different ways, but in child, it's really tough because you have mom reporting on her depression. You have mom reporting on how child is doing. You have mom reporting on parenting, right? And you often don't even have a secondary caregiver doing like a sort of confirmatory reporting. And we know that this is not the best basis for understanding a child's development. So what we do is behavior. It's so important to root our findings in observations in the children, but there's not so much that you can do in an infant in terms of behavior. Eye tracking is one of those really important behavioral avenues because you can provide a stimulus to the child. You don't have to solicit a response, but you can let the eyes track. There is development to be done in the eye tracking field. And again, this is not my field, so I want to be careful how that's my dog making noise. I want to be careful how much strength and how emphatic I am about the following statement. But even the variables that we extract in terms of look duration are really tricky because sometimes it's adaptive to have it's really only interesting when you have like two things on the screen versus an independent thing that they should be looking at. So just the kinds of analysis that folks do and the variables that we're plucking out of these data, I think there's room for growth. I think in terms of, what is it that you expect from an infant? It's an evolving field because with AI, we're going to change a lot of what we do with this eye tracking data.
No, I agree. In fact, we have one of our newest members of the Columbia community, Dima Amzo, is spending a lot of time with that, as you know, with her.
Well, so Denise is Dima's former grad student.
Right. Yes. Yeah. So thank goodness there's a little bit of this academic nepotism going on, or whatever. Or it's just, I think it's the Ridge Club network because we're all blessed with having these wonderful students being able to rotate through these labs. Let's see, Ezra just wanted to add that quick note. He really appreciated your caveats about what kind of results do get published. So, and then there is Robert Carter was asking, are you aware of fetal MRI work with prenatal alcohol exposure, focused on prenatal alcohol exposure itself?
Bill, we should collaborate.
Yeah. Yes.
I don't think there has.
I know of some where the study we did in South Africa, those kids were followed up at 11 years of age. And in fact, there's just a recent publication on that. I wish if I could think of the Dan Stein is I think is one of the authors, but I can't remember the other one. I can look into that and we'll send it to Robert. But yes, you're right. More needs to be done about that, and more needs to be done in the context of the other factors that we think are so. You can't just study alcohol, as you know.
Yeah, and Robert, if you do have interest in alcohol, another piece of this I think is that I recently was reading about is the comorbidity of like exposure to alcohol in utero with things like ADHD. It's a very high proportion.
Mm-hmm.
And how we haven't, I don't think as a field, really taken apart sort of familial ADHD, alcohol. And so we don't really know what is cause or consequence of these associations. And also we have a lot of children that are being diagnosed with ADHD that have prenatal alcohol exposure that aren't being studied separately from those with. So I think what this sort of just is something I've become aware of recently, is that we have a lot of murkiness in terms of these diagnostic groups that probably should be considered separately. So it's an interesting topic.
Yeah, no, I agree. And in fact, there was a related question on how it is that, given you can't do everything, but you have colleagues who are also collecting genetic material to understand how some certain genetic vulnerabilities can factor into the outcomes of interest.
Yeah, so you always run into sample size considerations with all of the really interesting things. Like right now we're doing metabolomics, right? In the new cases, we also have epigenetic data in the Detroit cohort at multiple time points. It's tricky, it's tricky. I'm one of those people who likes to see things replicated. I know we probably all are. So if you do have these really small samples and you want to look at these types of effects, and with neuroimaging, it commonly is, maybe the HBCD study is the answer to this question, as it will be one of the largest standardized imaging studies with that type of background. And actually, I should have just said that. I think HBCD, maybe the Developmental Connectome project. It's not something a single lab could do easily or well.
And hopefully they'll allow for some innovative measurements to go along with the HBCD too. We'll see how that goes. And the last question I have here in front of me anyway, it may be the second to last, but I think it's the last one. No, there's two more, sorry. This is by anonymous attendee. I'm not sure this is the best way to ask the question, but here goes. Do you see the changes induced by social environment on the fetal brain to be like brain injuries, or as organized responses, adaptations to adverse social environmental conditions, or maybe a combination?
Thank you, anonymous. I use the word adaptive a lot with my moms that are having babies during the pandemic. I'm calling their babies superheroes. I'm saying that their children are better adjusted to deal with the future, what it's like to live on this planet, than we are. The place where most of the researchers that work in trauma and stress tend to land, at least in my read of it, is that we are organisms that are highly evolved to adapt. That's just natural selection. We do really well if the future environment matches what we evolved or adapted toward. We don't do well if you put us in some sort of a situation and we have some sort of microevolution, and then you put us in an environment that's a mismatch. So usually when we think about trauma, we think about traumatic exposures, we sort of think, well, that child, although what they went through is really awful and terrible, maybe they're more primed for more vigilant responses in an environment that's going to continue to be hostile or challenging. It's the taking the child out of that environment where that hypervigilance is no longer adaptive that you have a problem. So when I talk to moms, that's my message. My message is very much like any differences that we do see are really your child's adaptiveness and ability to be flexible. Mm-hmm. And again, that's why, when you take something like THC use during pregnancy, coming in with a more neutral approach, not knowing if maybe that is having a therapeutic value, is a much better approach. And we have to be careful.
Great. So now this is the last question. I think Richard Brockman asked, and it relates to a couple of questions before. Does maternal depression with its associated high cortisol and decreased sleep, which is, you pointed out, occurs within 1 in 5 women, does that differentiate in its effect on network connectivity compared to other more external stressors? So.
I think maybe this is referring to things like lead, right? Is that what you think that that's like external toxicants?
Maybe that's correct.
Yeah.
Yeah.
So the proper way, I think, to study that is to do it within the same sample, right? And we don't have that right now. We don't have a very strong toxicant exposure sample to compare, like the psychosocial sample. There will be opportunities to do that in large scale studies, right? These small studies, it's a little bit trickier to do. We, with the tooth material that we'll be collecting and analyzing, will look at lead and other toxic metals, micronutrients, and inflammation over pregnancy. And we'll have measures over time, so we can look at sensitive windows and interactions across those. We'll use lag diffusion models. It's a rather complicated approach. And we will be able to begin to take apart sort of the external environment and the sort of the psychosocial, which we think of as more of an internal influence. But really, I think the best way to do this is with big studies. So we can begin maybe to generate hypotheses in these smaller samples, but to really begin to take apart those effects and the contributions of each to what we're observing, it's tricky. We have published a lead study. We have high lead rates in Michigan, so you can check that out. We did see what looked like less optimal development in those children that were exposed to lead in utero. We didn't make a direct comparison with maternal psychosocial health.
So I'm going to ask the very last question. So can you share with us how you managed to clone yourself so you could do all the wonderful, incredible?
That was not a question.
How did you do that? It's just so impressive. Yes, it is more of a comment, but we want to thank you again for not only everything you're doing and continuing to do and expanding into all these very timely and important questions that are facing our society right now. But I want to thank you also for being able to articulate it and share it with all of us and to get everybody else as enthused and excited about what you're doing as obviously you are. So thank you again, Moriah. This was just terrific.
Thank you. And I'm your neighbor, everybody. So please don't hesitate to reach out. Thank you, Bill.
Thank you.
Thanks.